So I'm here this morning with Jan Juvenot from N6 Tech. He's the director of product there, and today we're going to talk about PCR and avoiding over-amplification and general good things about genomics.
Thanks, Chris. I think half your audience already tuned out when you said PCR, but we're going to try to bring them back in because it's not as boring as they think it is. It's actually a lot of fun, but you got to stick around for the fun.
It's let's say it's not PCR like you're used to.
Exactly. Not your grandma's PCR.
I like it. All right, let's go for a little walk.
You know, it's funny because when we launched the project, when we came out of stealth at AGBT in 2024, that was basically Pranav's talk. You know, it was like introducing, hey, we're N6, and I know you guys are talking about a lot of cool things, but I want to talk about PCR. And it was super well received because, of course, it's like you go to AGBT and people are going there to talk about cutting edge technology and, you know, spatial transcriptomics and AI, and you come here and like, yeah, we're going to talk about a technology that's been around for 30 years.
Yeah.
So, I mean, bottom line, I think the philosophy behind is that there is no technology that's too mature to be revisited. Like anything, no matter how long it's been around, can be improved upon, and sometimes needs to be improved upon. So that's at bottom line what pushed N6 to develop Icon PCR.
Yeah, well, I think these folks are going to be blown away when they hear what's possible now with this. And so while we're walking over this way, let's talk about, for people who don't know about genomics workflows that aren't into it that deeply, what we're trying to do or what is the challenge with over-amplification and duplications and so on.
Absolutely. So everybody's familiar with next generation sequencing. It's really been the revolution of the 21st century in terms of biology. It has enabled so many discoveries. And the part that people don't talk about that much is what it takes to actually sequence a genome. And for most platforms, it does go through this process of library generation or library preparation, which involves oftentimes PCR. And I say oftentimes, it's like 95% of the time.
Yeah.
Most of the time, it involves a PCR step. And you need PCR because you're adding adapters, you're adding indices, you're using it just to generate enough material to actually get a meaningful sequencing. And for that, PCR is great. But it's also a necessary evil because the more PCR you do, the more errors, the more artifacts you introduce. And that's really the part that scientists who do NGS try to control. They try to make sure that they do just the right amount of cycles for their sample. Like if I have a very good quality sample, abundant in the micrograms, then I need very, very few cycles when I'm PCRing, maybe just when I'm amplifying, just 4 or 5. But if I start with cell-free DNA from a patient and I'm down in the sub-nanogram range and might be degraded, I'm gonna need, 15, maybe 20 cycles just to get a read on that patient. So those are the differences, and that's why it's important to know what the optimal number is.
All right, so we talked about the technology, or we talked about the problems. Now let's talk about the technology. So explain for people what the difference is in your platform and how you avoid those challenges of over-amplification and so on.
Absolutely. So if you're familiar with a PCR machine is basically a metal block with typically 96 wells where that has, Peltier elements underneath from 4 to 6 depending on the type of instrument. And those Peltier elements will heat and cool the metal block. And there's a lot of work that goes into the design of the metal block, the positioning of the Peltier elements, to make sure that all 96 wells are at as close to as possible to the same temperature every time. But those 96 wells will do the same thing. Like if you program them for 20 cycles, they'll all do 20 cycles. If you program them for 30, they'll all do 30. So that limits the number of things that you can do with a standard or legacy thermocycler. What N6 set up to do is basically, by understanding that PCR is this crucial step in NGS that doesn't really have a good solution, or at least not a good solution without a lot of manual intervention, I should say. We set off to design a smart thermocycler where instead of having a block, we have 96 heating elements that each have their temperature sensor tied to the well. And so basically it's 96 thermocyclers in the same footprint as a 96 well plate. And people ask, how come nobody thought of this before? I'm like, they might have, but it was a heck of a lot of work in engineering to miniaturize everything to fit in a 9 by 9 millimeter pitch. And that's really where the magic sauce is. It's making that happen and controlling the temperature at the millisecond level.
That's amazing. So what blows me away there is you could have one well that is done and being held at, I presume, a relatively low temperature,
yeah,
and whatever 6 wells around it, 8 wells around it, that are still going up and down, and it doesn't get heated by just the radiation surrounding it.
That's correct. So precisely when you're doing library prep and you've decided, you've set your level of fluorescence, you've spiked, of course, an intercalating chemistry into the mix so that you can monitor the synthesis of DNA. Once your D6 is done after 7 cycles, it will go down to 4 degrees or whatever cold temperature you set it up for, and everybody else continues cycling until each of them is done. And it's beautiful because you see that happening real time, and you're like, okay, we really got something here, because it's perfectly stable. Everybody else is not affected by the fact that there's a cooler neighbor next to them. Cooler neighbor sounds like something for a sitcom, but so
we all want cooler neighbors.
Exactly. This is the title of my next app note will be Cooler Neighbors. So, but yeah, no, that's really how it goes.
So let's talk about detection, which I think may be relatively common. Tell me if it isn't, by fluorescence. So you're watching the reaction in some way. That was not a thing when I was doing PCR. And then also a little bit about what the engineering took, like what was the breakthrough that allowed you to miniaturize and coordinate everything individually?
My engineers will probably kill me for speaking on their behalf, but I think really the design of the control and the boards that really synchronize all of this was where most of the work happened. Identifying what elements can generate enough power was one thing, and it was crucial, but really controlling them and synchronizing the reading of the temperature to the power is where a lot of the work went in, and doing that on a 96 level scale. So, yeah.
That makes sense. I didn't think of that, but like if you're talking about a large block and heating the whole thing, that's one kind of power distribution into it. But to deliver that much power locally through multiple, let's call them cables, I don't know what they are, but that's another challenge. Like,
it definitely is, and it's something that that I try not to get involved in, because it's funny, in the early stages of the company, and I was obviously one of the very early hires, I was trying to help with anything, and at that point we were working on the first prototype, and I told the engineers, guys, I need something to do biology with. What can I do to accelerate this? They're like, well, you can do inventory. So they set me off to count tiny pieces of components and transistors and all that stuff, and I'm like 160, 161, 162. But yeah, there are tiny elements. I mean, again, it's not done manually, other than for the prototype. But once it's automated, it was definitely a big relief.
Here's what I love about that story. I mean, we're talking about a legitimate technological breakthrough, but behind that is some not very pretty work of 1, 2, 3.
It was your typical tech startup origin story with garage sessions and screw ups, and it was made even more interesting by the pandemic because we were starting to design this instrument during the pandemic. So the meetings had to be held at a distance until we were all vaccinated and all that. So that was another challenge. But then after that, it got better.
You're the second company I've talked to about starting up in the pandemic where no one met each other. It was all remote during the formation of the company. That's pretty phenomenal. So one of the areas where you're having an impact, metagenomics. Mm-hmm. Carrie Hare at Penn State said, great discoveries are made at the margins. Where else do you see those little improvements having a big impact?
So metagenomics is definitely one of the fields where Icon PCR has been embraced the most organically, I should say, because it is a field that is really, really vulnerable to overamplification. And I'll tell you why. So the workhorse of metagenomics is the 16S sequencing. 16S is this gene that has a high diversity between species. Therefore, if you just sequence that gene, you can identify what species of bacteria you are dealing with. So when you're doing bacterial population analysis in soil or the waste residue or whatever, you want to be able to get as close as possible to the full picture. And the reality is that when you amplify those samples, the more you amplify, the more you increase the presence of what we call dominant species. So basically, species that are not represented very highly, but that have an amplicon with a PCR advantage. So in theory, every amplicon will be duplicated at each cycle. So you get one copy at cycle n, you get two copies at cycle n+1. That is in theory. There are some amplicons that have an advantage. They are more AT reach. They have less secondary structures. They're shorter sometimes. And so those amplicons will be closer to the maximum theoretical of 2. And other amplicons that may have like stem loop structures or high GC content, those might be closer to 1.8 or 1.6. So the more cycles you go, the less representation you'll have of those less favored amplicons and less favored species. So we've done this study with in partnership with Zymo and University of Arkansas, where we tested 64 different soil samples, and we compared the results on 3 different sequencing platforms using either the standard protocol or Icon PCR. And the number of species that are identified with Icon PCR is like sometimes up to 5 or 10-fold more than what you would get in the regular protocol. So that really is what Carrie meant when discoveries are done at the edge. It's those species that if you go 5 or 10 cycles more, you won't see them. So that is what scientists care about, is figuring out what else is going on in my sample that I'm not seeing.
All right, so looking down the road, how do you see this technology impacting things like liquid biopsies and preventative healthcare at scale?
Listen, I think anything that uses NGS will be impacted by this, because again, there is an enormous amount of progress being done on the sequencing capacity now. So sequencers now can do a lot more, gigabases than 5 years ago, but the process for sample prep, for library prep, hasn't evolved. So you're starting to hit this critical capacity where for liquid biopsy, it is now cost-wise to sequence to a certain depth and look for all these markers and try to detect all these types of cancers early. But as liquid biopsy becomes more and more the norm, the bottleneck is going to be your library prep and the types of methods that you use for that. So an instrument like the N6 Icon PCR helps not just streamline, but also increase the quality in the back end. Again, why is it important to use something like this? Well, if you look at the typical workflow for library prep, you're going to have, your adapter ligations, etc., then your PCR amplification. From there, you're going to have to purify individually all your libraries. You're going to have to quantify them individually, and then adjust the levels before you sequence them. So all those processes take time, use reagents, and they cost money.
And labor.
It's labor, exactly. So by using Icon PCR, what you're enabling is a process that we've called auto-normalization, or auto-norm. And you basically just say, hey, I want all my libraries at this threshold or at this condition. And once they reach there, they stop. And basically, by determining that threshold, you're ensuring that they're all pretty close in quantity, close enough that you don't need to quantify them. So basically, instead of purifying them all individually, you can just pool them, take 5 or 2 microliters of each of them, purify, put them in one tube, do that one purification instead of 96 individual ones, and then just put that product into the sequencer. That is a huge savings in terms of time and consumables. But more importantly, because now you have libraries that have not been over-amplified, what you are sequencing in the back end is going to have a lot less duplicates and a lot less artifacts. And there is a hidden cost to all this. Duplicates have to be removed, and all that raw data has to be stored. If you are paying to store crap, useless data, those are dollars that, okay, you may not see them because you budget overall, but they will cost you something. Storing garbage has a cost, and that's what we're trying to prevent. And as liquid biopsy becomes more and more popular, because let's face it, as much as you and I enjoy a colonoscopy, if you give me an option where it's just a blood draw instead of going to the gastroenterologist, I would much rather do that. So it's going to become the norm for not just cancers, but a lot of other conditions. And we want to enable diagnostic labs, clinical labs, to have options that are cost effective, that give them better quality and overall improve their workflow.
Yeah. What stands out to me there is the hidden cost of the things that seem free while you're doing them, like data storage, but in the end, they're not.
No.
Right?
No. There's, the concept of the cloud is nice and it's ethereal, but Amazon charges you.
Yeah.
And I mean, not just them. Anybody who gives you cloud storage will charge you for that. So yeah, be careful. Be mindful of what you put in there.
So this hull of a ship behind us was a lumber ship, and the captain of that ship, in his later years, decided to live on it and just beach it here.
Oh, wow.
So he lived here. And I don't know when he passed away, but the ship eventually burned down to the water line, as you can see here in like 1976, so not that long ago. But that ship's been here for many, many years, and it was previously somebody's home.
That's very cool.
Yeah.
I'm sure he didn't have a permit to live here, but he's like, you know what?
There's
Sue
probably
me.
no permits back then.
Exactly.
You do whatever you want. The law of the sea.
Exactly. He's a pirate after all.
Yeah. A pirate. All right. So thinking 5 and 10 years ahead, what things in genomics are you most excited about, and how does this all fit in?
I mean, definitely healthcare wise, I am excited by the type of discoveries that liquid biopsies are going to be enabling. I think there are ranges of diseases that can only be detected too late right now. And that is definitely something that, you know, NGS is going to make possible. On the more basic side, I am very excited by the fact that now we have tools that allow us to study nature and biology in all its complexity. Like, you don't have to settle for a model, you don't have to settle for a couple of genes and see how, you know, increasing one affects the other, etc. You can have a holistic view of your entire system, and that is crucial. And again, I mean, I'm sure my biochemist colleagues are going to be like, "And don't forget about the protein," and I'm not forgetting about the protein. But right now, protein sequencing is not a reality. So until then, but yeah, there's a whole lot of new perspectives that are going to be open just because we now have this global picture. I mean, it's, to give you a metaphor, like if aliens were observing this planet and they decided to choose just one marker to understand, you know, human life, and the marker was chairs or, you know, cars, and it would be like, okay, we're observing this one marker and we see movement, and when we see these chairs, they sometimes are static, but at certain regular hours they come out and then they come in, and you try to establish a model based on just one marker, you're bound to get it wrong. You're bound to not have the whole picture. So that's why any tool that allows you to observe the entirety of your system is bound to give you much better comprehension of how it works. 5-10 years from now, I really hope we have embraced that on many different levels. Just, you know, even just environmentally, like when we are looking at species, you know, in the past, did we care that, you know, yeah, we cared that pandas were, you know, the panda population was going down, like, and that's great, that's important. They, you know, they shouldn't disappear. But like, did we care about everything else? Like, now we have ways to see, give me panda poop, and I'll tell you what else is going wrong in their environment, what else is disappearing, what else is causing this population to go down. Panda is probably not the best example because they're really well characterized, but any other species, you know, like that, you can now have a vision that is a lot more encompassing that we used to have before.
Yeah, so that just brings up a thing, you know, I was just looking at yesterday. I'm trying to remember what it was. It was a discovery, one of those long ago discoveries in basic research that no one would have ever thought would have an application, and then years later, and maybe it'll come to me, what we use that discovery for now. It had something to do, I think, with some rare animal or whatever. You know, like when we discovered Taq polymerase, right? I mean, that was just basic research, and then has turned into an industry in itself.
It's always like that. It's always, oh, this is not applied science. I'm like, applied science has to start from basic science. And if you only look for things that have concrete applications right now, you're going to block yourself from solving the problems that come much later. And that's really something that, I mean, as a society, I don't know how much N6 can do about it, but as a society, we need to embrace. It's just necessary, constantly, you know, a certain percentage of any research budget needs to go for that, because otherwise you don't have any progress.
Yeah, we have to keep working on that. And the other thing that stood out to me, that when you talk about the protein biochemist, it's like when I was interviewing for graduate school, the guy who ended up being my advisor, John Roth, he said, I remember sitting down in his conference room, and the first thing he said to me is, we do genetics as a business here, and we only do biochemistry when we know what the answer is going to be. Which is never, you know, that was whatever, 40-some years ago, but I've never forgotten that.
Having lost a lot of hair over pouring Western blot gels, I totally understand that, and I empathize with it. I couldn't. I mean, I know why, and I know there are other tools, by the way, biochemists, don't hate on me. I know you've made a lot of progress.
Leave your comments below, address them to Jan at.
Like, hashtag protein silencing.
All right, last question. You talked to me previously about, you know, postdocs telling you their stories of having to watch their reactions, you know, late at night, going, is it done yet? Or when do I stop it?
This is the beautiful part about the solution like N6 is like, listen, I can tell you, I can break down in, you know, dollars and hundreds of dollars how much your lab is going to save and everything. But to me, something that speaks a lot more is, yes, postdocs, which, by the way, just as recently as last week, I had one who told me that that was exactly her life, who have to watch their library amplification on their regular thermocycler. And they have spiked fiber in it. And once it reaches a certain point, they pause the thermocycler, take out that one tube, and continue cycling for the rest. And they have to do that for the entire reaction. And, you know, it reminded me of those old-timey PCR people that told you, oh my God, you whippersnappers just put your plate in and press 40 cycles and off you go. I had to like transfer my plate from water bath to water bath, you know, holding a timer, and that lasted 2 hours, etc. Well, it's the same kind of progress that we are nailing right now. Like that postdoc that has to watch her libraries go, I mean, her time is free now. Just program your auto norm, walk away, go to the rock climbing gym, go write some poetry, go take a nap. You now have a lot more time to yourself. And this is, you know, again, quality of scientist life is not a topic that is brought up too often, but having been on the receiving end of that equation, I know how many stupid hours are wasted because of that, because there is no better way to do it. And we're really happy to be able to do something about that too.
Yeah, you think about working in a, let's call it a very high-tech environment, and yet being limited by manual labor that doesn't exist in many other lower tech places, right? I mean,
yeah, no. Listen, I look forward to receiving this letter a year or two from now from some postdoc who is thanking us because with all the free time that we gave her, she managed to write her first album, and now we have the new Phoebe Bridgers or, you know, Chapel Roan, because these were hours I didn't have before, and now you're giving them back to me. So thank you. I don't know that will be the case, but in my dreams, that's what I look.
I like it. Yeah. Well, Icon Jan, thank you so much. This has been a blast.
It's been great, Chris. Thank you for having me.
My pleasure.